Welcome to Chapter 10 of the Arborist Exam Prep, Diagnosis and Plant Disorders. Diagnosing plant disorders requires a combination of knowledge, experience, observation, and deductive reasoning. A disorder impairs a tree's vital functions, and insects and pathogens often target trees that are already stressed or weakened. The diagnostic process involves two essential steps: identifying the cause of the disorder and considering treatment options and making informed recommendations. And since trees can't describe their symptoms, diagnosis depends on gathering information from the site and client observations. Key questions to ask the client include: how long has this issue persisted? Were there any early symptoms? Has there been construction, excavation, or chemical treatment nearby? It's important to remember that the information provided may not always be accurate, so cross-checking is essential. Tree disorders fall into two broad categories. You have your biotic factors, or living, and your abiotic factors, which are non-living. Biotic factors include fungi, bacteria, viruses, nematodes, parasitic plants, insects, mites, and other animals. Pathogens spread between plants and are considered infectious. Abiotic factors include mechanical injury, such as from equipment or weather events, environmental factors such as temperature extremes or moisture fluctuations, and soil-related issues, including compaction and mineral deficiencies. It's important to learn the difference between symptoms and signs. Symptoms are the plant's response to the stress or damage, such as chlorosis or yellowing of the leaves, wilting, and leaf scourge. Many disorders share similar symptoms, complicating diagnosis. Signs are direct evidence of the causal agent, such as fungal fruiting bodies, insect frass, or exit holes. Identifying signs helps distinguish between primary and secondary causes. Recognizing symptoms and signs helps narrow down potential causes. We're going to go through some of the most common you may run into in the field. First, we have blight, which is defined as rapid death of flowers, leaves, or stems. Blotch, which is irregular dead areas on plant tissues. Canker is localized dead areas on stems or roots. Chlorosis is yellowing due to the lack of chlorophyll. Decay is wood decomposition from fungal activity. Dieback is progressive death of the branches. Gall is abnormal growth due to parasitic activity. Gummosis is sap exudation from disease or injury. Necrosis is death of plant tissue. Oozing is sap or liquid exuding from the wounds. Scorch is browning along the leaf margins. Leaf spot is localized dead areas on leaves or stems. Stippling, speckled areas from insect feeding. See mite damage. Stunting is reduced growth from stress. Vascular discoloration is darkening of the xylem and phloem due to disease. Many plant disorders display nonspecific symptoms, making diagnosis challenging. To reduce errors, first we need to analyze tree history and environmental conditions. Consider multiple factors before drawing conclusions, because misdiagnosis can lead to ineffective treatment and wasted resources. The first step in any diagnosis process is correctly identifying the plant. This limits the list of possible causes because many pests and pathogens target specific species. It's essential to recognize species, hybrids, and cultivars and their susceptibility to biotic and abiotic stresses. It's important to examine the pattern of damage to identify possible causes. Non-uniform damage suggests biotic factors, living. Uniform damage often indicates non-living or abiotic causes. A large-scale, uniform pattern usually points to non-living stress factors. Site evaluation is the next step in the diagnosis process. You want to consider light, soil quality, water availability, and wind exposure. Look for recent construction, soil compaction, or proximity to roads or de-icing salts. Comparing affected trees to healthy ones can provide useful clues. Abnormalities in the foliage often indicate root or vascular issues. Key symptoms include chlorosis, which is often linked to nutrient deficiencies or root stress. Twisting or curling may result from viral infections or herbicide exposure. Early fall color is a sign of root girdling or chronic stress. Examine the trunk and branches closely. Look for a discoloration, oozing, and loose bark. Check for pest entry points such as emergence holes. Excessive water sprout growth often indicates tree stress. Measuring the terminal bud scale scars can help evaluate growth over time. Patterns to watch for are slow growths, which is often caused by chronic site issues or climate factors, or a sudden reduction in growth, which could indicate construction damage, soil addition over roots, or defoliation. The tree's root system is often overlooked in diagnosis. You want to check for decay in major roots in the root collar, soil texture, moisture, and odor. Healthy roots should be light-colored, firm, and flexible. You want to look for dry roots, which are brown and shriveled, or waterlogged roots, which could be black with a sour odor. Common root stress factors include soil compaction, which limits oxygen and water uptake, poor drainage, which creates an anaerobic condition, chemical exposure, damage from de-icing salts or herbicides, and girdling roots, which restrict vascular flow leading to decline. To help narrow down the cause, you want to eliminate the possibilities one by one. 70 to 90% of plant problems are due to abiotic environmental factors or poor site conditions. Opportunistic pathogens often attack stressed trees, complicating diagnosis. Abiotic disorders commonly result from drought, overwatering, soil compaction, nutrient deficiencies, temperature extremes, and mechanical injuries. And biotic disorders involve fungal diseases like root rot and cankers, bacterial infections causing wilt or galls, viruses that lead to stunting or abnormal growth, insect pests such as borers and defoliators, nematodes, and parasitic plants. As I mentioned, abiotic problems often show up as uniform symptoms across multiple species. Site-wide damage is linked to environmental stressors. Check for recent construction, watering practices, or soil contamination. And we also mentioned biotic problems usually cause non-uniform damage within the same species. You want to look for visible signs such as frass, exit holes, or fruiting bodies. Discoloration and dieback can indicate biotic stress as well. Correct diagnosis guides effective treatment. You want to focus on improving overall tree health, addressing abiotic stress first, managing biotic factors with appropriate treatments such as fungicides, insecticides, or pruning as needed. Tree decline is often a slow process influenced by multiple factors. Predisposing factors are age, genetics, and poor site conditions. Inciting factors are defoliation, frost, or drought, and contributing factors are secondary invaders like borers or fungi. Identifying all contributing factors is essential for an accurate diagnosis. Symptoms of decline develop over many years. Common indicators include reduced growth, discolored leaves, and canopy dieback. Early intervention can sometimes reverse the decline. Proper diagnosis involves evaluating multiple factors. Many pests and fungi are secondary invaders that target stressed trees. If you're unsure, seek expert advice. Guessing may result in ineffective management. Tree stress falls into two categories: acute stress, which is sudden events such as lightning, frost, or chemical injury, and then you have chronic stress, which is long-term damage caused by poor soil, drought, or nutrient deficiencies. Symptoms may not appear immediately, which delays diagnosis. Abiotic factors typically affect water and nutrient uptake, often resulting in delayed symptoms. Common factors include poor soil conditions, mechanical injury, and extreme weather events. Poor drainage, soil compaction, or incorrect pH can be abiotic factors. These problems lead to crown symptoms such as dieback and discolored foliage. Assess soil and root conditions before diagnosing. Physical damage from lawnmowers, string trimmers, or vandalism can cause decay, structural instability, and dieback over time. Mulching around trees helps prevent mower damage. Extreme weather can severely impact tree health. Frost crack, sun scald, wind throw, and stem failure are common. Choosing appropriate species for the site helps reduce susceptibility. Trees often compete with surrounding plants for water, nutrients, and light. Shade-intolerant trees may experience lower branch dieback and reduced growth. Allelopathy occurs when some trees release chemicals that inhibit the growth of nearby plants. Herbicide drift or misapplication can harm trees, causing symptoms that mimic nutrient deficiencies or pest damage. Common symptoms include leaf curling, discoloration, and leaf drop. Water stress causes a range of symptoms. Drought leads to smaller leaves, leaf scorch, and premature defoliation. Excess water can lead to root suffocation, decay, and anaerobic fermentation. Prolonged water stress attracts opportunistic pests and pathogens. Girdling roots constrict vascular tissues, limiting nutrient flow. Common causes of this include improper planting depth and twine left over from balled and burlap nursery stock. Prevention involves inspecting nursery stock for circling roots and ensuring proper planting depth. Many different insect species can be found on a tree. While some may be harmful, many insects provide benefits by preying on pests. It's important to remember that most trees will have at least one insect pest capable of causing problems. We can divide these insects into two broad categories. There's general feeders and host-specific pests. General feeders such as Japanese beetle, aphids, and gypsy moths feed on a variety of tree species. In contrast, host-specific pests like emerald ash borers target specific species. Recognizing these differences is key to managing insect populations effectively. Correctly identifying insects is critical to managing tree health. Understanding their life cycles helps determine whether the intervention is even necessary. Some developmental stages may be harmless, while others can cause extensive damage. For example, caterpillars of certain moths defoliate leaves, while adult moths do not cause any harm. Proper identification allows us to assess the threat level and decide if controlled measures are warranted. When in doubt, it's best to consult a diagnostic lab or entomologist to ensure accurate diagnosis. Most insect damage to trees results from feeding or egg-laying activities. One example is cicadas, which lay their eggs in slits they create under the bark and small branches. This weakens branches, making them prone to breakage. During high cicada population years, this damage can be severe, especially for young or newly planted trees. Understanding these types of damage helps us assess whether intervention is even necessary. Chewing insects are a diverse group that includes caterpillars, webworms, sawfly larvae, beetles, and weevils. They consume leaves and stems, causing various types of damage. For instance, gypsy moths and cankerworms cause defoliation, stripping trees of their foliage. Black vine weevils feed on leaf margins, leaving notched edges. Japanese beetles and elm leaf beetles skeletonize leaves by consuming tissues between the veins. Recognizing these damage patterns helps in identifying the pest responsible and determining the appropriate course of action. Leaf miners and bark beetles are two groups of insects that cause internal damage. Leaf miners tunnel between leaf surfaces, creating distinct patterns. Bark beetles and borers, on the other hand, lay their eggs under the bark or on the tree's surface. Their larvae feed on the phloem, cambium, and xylem, disrupting the tree's nutrient and water flow. Symptoms of bark beetle infestation include crown thinning, wilting, chlorosis, and branch death. Identifying these signs early is crucial to preventing significant damage. Piercing sucking insects include aphids, scales, leafhoppers, psyllids, and true bugs. They feed by piercing plant tissues and extracting fluids, which can cause chlorosis, stippling, leaf drooping, and distortion. Some of these insects also inject phytotoxic chemicals during feeding, leading to further damage. While a small population may not cause immediate harm, severe infestations can stress and weaken trees over time, making them more susceptible to other pests and diseases. Certain insects such as aphids, soft scale, and mealybugs excrete a sugary substance known as honeydew while feeding on phloem sap. This honeydew coats leaves and branches, creating a favorable environment for sooty mold fungi to grow. While sooty mold is primarily an aesthetic issue and does not penetrate plant tissue, it can reduce photosynthesis if it becomes severe. Over time, heavy infestations of honeydew-producing insects may lead to a decline in tree health. Some insects act as vectors, spreading plant pathogens and increasing tree vulnerability. Bark beetles, for example, transmit the fungus responsible for Dutch elm disease. Fire blight bacteria are often spread by bees, and aphids and leafhoppers can transmit viruses. Managing these insect populations not only protects the tree from feeding damage but also helps prevent the spread of harmful diseases. The timing of defoliation significantly affects tree health. Early season defoliation is more harmful because it depletes carbohydrate reserves needed for growth and defense. Late season defoliation, although visually striking, is generally less damaging since the tree has already stored sufficient reserves. However, evergreen conifers are especially vulnerable because they do not readily regenerate foliage, making them more susceptible to long-term damage from defoliation. Mites, though small, can cause significant damage to trees. As arachnids related to spiders and ticks, they feed on plant tissues, leading to various symptoms. Spider mites, for example, extract leaf fluids, causing leaf discoloration and stippling. Areophyid mites induce abnormal growths such as galls on foliage and twigs. Some mites are predatory and can help control other mite populations, contributing to natural pest control. That's why identifying the type of mite and assessing its impact is important for determining whether treatment is needed. Nematodes are microscopic, unsegmented worm-like organisms that attack plant roots and tissues. They can cause a range of symptoms, including galls, swelling, chlorosis, stunting, and wilting. Root-feeding nematodes disrupt the plant's ability to absorb water and nutrients, leading to long-term stress. However, not all nematodes are harmful. Some species are beneficial and are used as biological controls against soil-dwelling insect pests. Proper diagnosis is key to distinguishing between harmful and beneficial nematodes. In addition to insects and nematodes, various animals can damage trees by girdling stems, which disrupts transport of water and nutrients. Woodpeckers peck at the bark in search for insects or sap, sometimes creating extensive damage. Deer browse on buds, twigs, and leaves, deforming young leaves. Squirrels may chew bark and clip twigs, particularly during winter, when other food sources are scarce. And rodents and small mammals can cause serious damage to trees, especially during winter. Rabbits, mice, and voles gnaw on the bark and clip off twigs, often girdling stems and causing death. Winter damage is more severe when snow covers the ground, limiting their food availability. To prevent such damage, avoid using coarse mulch near the tree base, which can attract rodents. Installing plastic trunk guards provides additional protection by preventing rodents from gnawing on the bark. Let's talk about tree diseases. Tree diseases occur when three conditions are met: a susceptible host, the presence of a pathogen, and suitable environmental conditions. This relationship forms the plant disease triangle. Over time, these factors interact, and the addition of time transforms the triangle into a plant disease pyramid. Pathogens vary in their host range. Some are host-specific, affecting only certain species, genera, or families, while others have a broad host range. The severity of the disease depends on which part of the tree is affected. For instance, vascular tissue infections such as Dutch elm disease or oak wilt often lead to rapid tree decline and death. Understanding the relationship between host specificity and disease severity helps guide appropriate treatment decisions. Foliar diseases primarily affect leaves, fruit, and young twigs. While these diseases are often an aesthetic concern, repeated defoliation weakens trees over time, making them more vulnerable to other stresses. Accurate diagnosis often requires microscopic examination of spores. Prevention strategies include planting resistant varieties and avoiding prolonged leaf moisture, which causes favorable conditions for fungal development. Shoot and twig blights cause wilting and necrosis of leaves and stems. Cankers often form on affected shoots and twigs, which can lead to dieback. These diseases are primarily caused by fungi and sometimes by bacteria. Trees stressed by environmental factors are more susceptible to shoot and twig blights, emphasizing the importance of maintaining tree vigor to prevent infection. Vascular wilt diseases disrupt water and nutrient flow by infecting the xylem and phloem. They typically enter through the wounds caused by pruning, vector feeding, or grafted roots. Notable examples include Dutch elm disease and oak wilt. While vascular discoloration is often visible, lab testing may be required for accurate diagnosis. Prompt action is necessary to prevent the spread of these aggressive diseases. Root diseases attack both fine feeder roots and large woody roots. Symptoms often include reduced growth, wilting, dieback, and crown thinning. Unfortunately, crown symptoms may not develop until significant root damage has already occurred, making early detection difficult. Proper site assessment and monitoring are essential for managing root diseases effectively. Rust fungi require alternate hosts to complete their life cycle. They attack leaves, fruits, and stems, often causing significant damage. Management strategies include planting resistant varieties, improving overall tree health, and avoiding planting of alternate hosts nearby. Preventing the introduction of rust fungi helps protect tree populations from recurring infections. Fungi play a dual role in tree health. While most diseases are caused by fungal pathogens, some fungi, such as mycorrhizae, are beneficial. Mycorrhizal fungi form symbiotic relationships with tree roots, aiding in nutrient uptake and improving tree health. However, fungal pathogens thrive in moist conditions and specific temperatures, making environmental management a key factor in preventing infections. Bacterial diseases present unique challenges for tree management. Common examples include fire blight, which is spread by insects, bacterial leaf scourge, vectored by insects as well, and crown gall caused by soil-borne bacterium. Temperature and moisture conditions significantly influence bacterial spread, making cultural practices and environmental management important. Phytoplasmas are bacterial-like organisms that live in phloem tissue and cause yellows diseases, such as elm yellows. Viruses, on the other hand, are spread by insect vectors and can cause symptoms like ring spots, mosaics, and mottling on leaves. Unfortunately, viral diseases often lack effective treatment options, emphasizing the importance of prevention and vector control. Few curative treatments exist for most tree diseases, making prevention the best strategy. Planting resistant species, supplying fungicides or antibiotics as preventative measures, and improving cultural conditions to enhance tree health are all effective ways to reduce disease incidence. Healthy trees are better equipped to withstand pathogen attacks. Global commerce has increased the spread of non-native species, leading to the introduction of invasive pests and pathogens. Native trees often lack resistance to these exotic threats, allowing them to spread rapidly and cause widespread damage. Effective management includes implementing inspection protocols, establishing quarantines, and supporting eradication efforts. Biological control involves introducing predators or pathogens from a pest's native range to control invasive species. While this approach can be effective, introducing new species carries ecological risks. Researchers and policymakers carefully evaluate these risks before proceeding to ensure that introduced species do not cause unintended harm to native ecosystems. Diagnostic laboratories play a critical role in identifying pests and pathogens, culturing fungi, and recommending management strategies. Tools such as microscopes, pathogen culturing techniques, and advanced RNA-DNA analysis allow for precise identification of causal agents. Submitting samples to diagnostic labs ensures accurate diagnosis and informed management decisions. Proper sample collection is key for accurate diagnosis. Samples should represent affected tissues with a clear interface between healthy and diseased areas. Leaves and branches should be freshly collected and wrapped in dry paper towels, while mushrooms and fruiting bodies should be placed in dry paper bags or boxes. Correct sample handling helps preserve diagnostic quality. Samples should be packed and shipped early in the week to avoid delays. Using the fastest shipping method helps prevent deterioration during transit. Detailed description of the host plant, site conditions, and any available photographs should accompany the samples to provide context for diagnostic lab technicians. These steps ensure the best possible analysis and recommendations. There you go. There's chapter 10 in the books. Thanks for watching. Hit like and subscribe. Chapter 11 coming soon.